Today we'll learn about enzymes. Enzymes are a type of protein which we covered in the last class. And last class we also looked at this table which includes some of the many jobs of proteins, and one of those jobs is this one. Some proteins are enzymes, for example, your digestive enzymes. And enzymes speed up chemical reactions. They are so important that without them, chemical reactions would be too slow to sustain life. Since they are so important, we're going to take a nice and close look at enzymes. Enzymes control the rate of reactions by lowering the activation energy needed to get a reaction started. Activation energy is the energy needed to start that reaction. So once a reaction is up and running, it can complete. However, it takes a huge amount of energy just to get it started. It's like you in the morning. Let's say it's a really nice Sunday and it's great weather outside and you could go outside for a run or hang out with friends, and there's all these things you can do. But first, you have to make yourself get up out of bed, and that's difficult. It takes a lot of energy to make yourself throw off the covers and get up. That is activation energy. And enzymes in reactions lower that amount of activation energy. Just like, let's say you woke up one morning and it smelled like cinnamon buns baking in the oven. That would lower your activation energy, and it would probably be easier for you to get up out of bed, and it would probably be a lot quicker too. So here is a chart that shows you how much energy is needed with and without enzymes. So here are some reactants, and we want them to turn into this product. And what you see here in the lime green shows you how much energy you need to get the reaction started if there are no enzymes. So this huge hump is the energy you need just to start the reaction. Once you get over that hump, the reaction happens. Now, with the use of enzymes, you need a whole lot less energy, smaller bump, and you can get from the reactants to the products much faster because you're not waiting around to get all of that activation energy. That's how enzymes work. So enzymes help in all different types of chemical reactions, but we'll talk about two main ones. So first of all, we have reactions like this one over here, where you're trying to take two monomers and build a polymer. This is a synthesis reaction. You're building something. The reaction on the bottom shows you the opposite, where you start with something like this could be a disaccharide, and you're breaking it apart into two monomers. This is like hydrolysis, right? Or this might happen in your digestive system. So enzymes help with both of these processes. In the first example, an enzyme will actually help to grab and find this yellow monomer and this red monomer, and it will hold them together and help them react with each other. It will catalyze the reaction so that we can create this product. Now, another important thing to note is when enzymes are involved, we don't call these reactants, we call them substrates. That's a special term that we use in enzyme catalyzed reactions. Now, enzymes also help break stuff apart, and you're trying to do just that in this chemical reaction on the bottom. And in this case, the enzyme would grab, let's say this could be maltose, right, which is a disaccharide. And an enzyme will grab the maltose, and it will actually help to cut the bond between the two monomers and help them separate and form into two separate sugars in this case. So that's how enzymes work. Now let's take a closer look because the enzyme isn't drawn in on this picture. Okay, so here we have a picture showing you enzymes, and the enzyme is here in this pink color, and this is the substrate that we want to perform the reaction on, and in this case it's maltose, which is found in Whopper candies, which don't taste very good unless you like them, in which case they, I guess, taste very good to you. But anyways, the enzyme has a really weird shape, and that shape allows it to fit perfectly with that substrate. So you can see that this can actually fit in here. Now, the enzyme has a region known as the active site, and the active site is the part that the substrate fits into. So it's this part of the enzyme, and you can see it here. It's this part of the enzyme. This is called the active site. And when the maltose binds to the active site of the enzyme, we call them together the enzyme substrate complex because it's made up of an enzyme and a substrate. So once it's bound, What happens here is the enzyme is going to help to break these bonds between these two glucose monomers, and then the product is going to be released. In this case, two glucose monomers are off to go and do their thing. Now, the important piece that you need to know about this is that enzymes are not used up in the reaction. They are left unchanged. So at the end of this reaction, the enzyme is free to grab another maltose and repeat the reaction, and then let the two glucose monomers go and then grab another maltose, and it's going to keep doing this. This is another example. In this one, we're doing the opposite. We're not trying to break something apart. We're trying to join things together. So here's your enzyme. It looks like this purple jelly bean, and it's going to reach out and it's going to grab this yellow substrate and this red substrate. This is the active site of the enzyme, and when the two substrates bond with the enzyme, it's called the enzyme substrate complex. And what you can see here, which we didn't see in the other picture because that was a simpler picture, is that the enzyme actually isn't a perfect fit in the beginning for the two substrates, but here it kind of changes shape a little bit and it molds and fits in with the substrate perfectly. When this happens, the enzyme is going to help the reaction between the yellow and the red molecule take place, and the product is released. Now, the enzyme goes back to its original shape. It's not warped or bent or damaged, and it's free to do more of the same type of reactions. There are thousands of types of enzymes inside of your body, and each one is specific to one reaction because of its unique shape.